So, hello everyone. Sally has kindly asked me to moderate today's webinar. My name is Michelle and I'm from the Advanced Molecular Diagnostics Laboratory in Toronto, Canada. I'm one of the molecular technologists in the lab, and we're affiliated with the University Health Network in Toronto. So for those of you who are not AGT members, please consider joining the association as we try to continue to be the voice of the technologists in the laboratory community and offer programs like this in the future. So please check out AGT's website, www.agt/info.org, and to see the benefits of memberships. So a few quick housekeeping notes before we get to today's presentation. Please feel free to ask questions during the session. In the upper right side of your computer screen, you'll see a control panel. In the lower portion of that panel, you can type in a question or comment and submit it to the webinar organizers. You can do this at any time during the presentation. We will reserve about 10 minutes at the end of the presentation for any Q&As. If you're participating as a group, please designate one person in the group to complete the online evaluation. In order for everyone to receive their certificates of attendance, that person will need to enter the names and email addresses of each person who is participating at your site today. As a backup measure, a sign-in form can be completed and faxed to AGT as well. Certificates should be mailed to participants approximately 30 days following the webinar. So I think we're good to begin. So today, quite lucky to have Dr. Miriam Thomas-Matthew, who will discuss the integration of technical bioinformatic and variant assessment approaches in the validation of a targeted next-generation sequencing panel for myeloid malignancies. Dr. Thomas is a postdoctoral fellow working with Dr. Suzanne Kamau-Reed and Dr. Tracy Stockley in the Variant Interpretation Group at the Princess Margaret Cancer Center. The focus in the lab has been the analysis and implementation of NGS panels and the interpretation of molecular variants in the context of cancers and other inherited disorders. So today we'll present Dr. Thomas.
Thank you, Michelle, and I'm very happy to be here today to talk about some of our considerations while we were looking to validate this commercially available NGS panel in our lab here in Canada. So yeah, I'm looking forward to taking you through our experiences here. So just a bit about molecular profiling in heme malignancies. The current molecular genetic testing environment here, at least in Canada and in many other parts as well, is focused mostly on single gene approaches where individual genes are tested for their usefulness in terms of diagnostic, prognostic, or predictive biomarkers. But recently, there are a number of new findings that have increased the number of these genes and these variants that are of clinical relevance and therapeutic relevance, which makes it really important to move into an area where you can more efficiently assess these multiple markers. And so that was our goal in setting up this validation, setting up this study that I'll go into, with the eventual goal of patient management. More accurate patient stratification using multiple gene markers will allow the physician then to improve clinical management and deliver more personalized treatment options, which eventually our hope is will improve patient survival. And so the current state of molecular testing for myeloid malignancies in the Princess Margaret Cancer Center and the Toronto General, which are part of the University Health Network for AMLs, the common testing methods include testing for the nucleophosmin gene, NPM1, internal tandem duplications in the FLT3 genes, and exons 8 and 17 of the KIT genes. And the methods are indicated on the slide as well. Currently for MDS, there is no routine testing, and we're hoping that this will change given the results from our study. And MPNs, again, are individual gene assessment for JAK2 and exon 14, for the CALR gene, and also JAK2 and exon 12. So these are the clinically relevant targets that are known so far and that are being tested. Other genes that are tested also include the ABL1, specifically tyrosine kinase domain containing mutations. And also the MDS-MPN overlap syndromes currently have no testing available. And so the panel that we've decided to test in the lab is a commercially available panel from Illumina. And you can see the genes listed here. It's a 54 gene panel, which includes coverage in hotspot regions for 39 of these genes, and complete gene coverage or complete exon coverage for 15 genes. And these are listed in blue on this slide here. Just a very quick overview of our validation methods. I'm not really going to go into our complete validation, but really just touch upon areas that we felt were important as we assessed the data that was coming out from the validation, and considerations that we used, which hopefully you'd be able to also take away and apply to different assays here. We used NGS library preparation and sequencing from the Illumina MySeq reagent kit. This was version 3 that we used. Our read lengths were paired reads of 250 base pair in size. We tested a number of samples as part of our validation, including the known HapMap samples that have been profiled previously, and there's good data available on the variants identified in these samples. These are NA12878 and NA12040, and also samples from a range of heme malignancies that we expected to be tested on our platform, including those outlined here. Our data analysis and interpretation was done using a combination of methods, including the commercially available NextGene software system, as well as the MySeq reporter software system that's included within the Illumina MySeq instrument itself. We have in our lab custom generated an informatic algorithm for detection of a large deletion in CALR. So this is a recurrent deletion of 52 base pair in size that occurs in CALR, and this is known. This is a known marker that appears in myeloproliferative neoplasms, or MPNs. We wanted to make sure that the methods that we're using are able to effectively capture this large deletion, and so in addition to the commercially available analysis methods, we've also designed our own to make sure we're able to capture this effectively. And we've also used Cartagena Bench Lab NGS for our variant prioritization and variant assessment needs, and I'll go through some of this as we go ahead. So just some considerations during our validation and the thoughts as we went into implementing our panel for myeloid malignancies. We wanted to make sure that we're able to capture clinically relevant or technically challenging genomic regions that are known in myeloid malignancies, and if we can't, that we effectively find ways to address any limitations of the system. And so there are 3 in specific that I'll talk about today. One is the CBP alpha gene, which is known to have high GC content and therefore poses a challenge in sequencing. The FLT3, which is known to contain large-sized insertions. Typically, it's identified by our FLPs in the routine lab, and so we wanted to understand how well are we able to capture this by NGS using the panel that we have. And also CALR, I mentioned a little bit about this already, but this is mostly an informatics question of whether 52 base pair recurrent deletions can be picked up. We also looked at sample type considerations. We expected to receive both blood and bone marrow as part of our test, and so we have data showing how these 2 sample types performed in the assay. I also share with you our considerations for confirmatory testing. I know different labs have different algorithms and different means of Sanger testing or testing using an orthogonal method. We'll discuss some of ours. And finally, how we prioritize and how we evaluate our variants to assess them for clinical relevance. So starting with CBP alpha, so this is a transcription factor involved in myeloid malignancies, and recent advances in the field have defined biallelic CBP alpha mutations as being prognostically significant in AML. This, in fact, is considered to be a separate disease entity when you have two mutations in CBP alpha, and this particular subtype is known to have a better prognosis compared to the rest of AMLs that have either wild type or other variant containing AMLs. And so identifying variants in CBP alpha becomes very important clinically in our system. But we also know that it is associated with high GC content. Over 75% of its coding region within the CBP alpha gene is high GC, and so we wanted to assess in terms of capturing this region and amplifying this region how well it was doing in our assay. So just to show you here on the top bar, you'll see the CBP alpha coding region, and this is covered in the panel by 6 amplicons, as I've shown here, amplicon 1.1 all the way to 6.1. We assessed the performance of each one of these amplicons in terms of how well each amplicon is covered in our assay. And so you can see on the bottom left graph the average depth of coverage for each one of these 6 amplicons covering codons indicated on the x-axis. And you can see that for 5 of these 6 amplicons have coverage with a median coverage of less than 500x, and usually 500x is our threshold for defining a failed region, and so anything below 500x is something that we would need to supplement by either Sanger sequencing or orthogonal methods that would allow us to fully interrogate that region. The last amplicon seems to work really well, but unfortunately for the majority of this region, sequencing did not meet our 500x threshold. And so we also wanted to make sure that these regions also contained relevant mutations. And so we looked within the COSMIC database, which is a repository of all somatically acquired mutations. And so you can see here the number of variants reported in COSMIC. There are a significant number of mutations lying within regions covered by amplicons 1 through 5. And so we would, in fact, be missing mutations without supplementing this region. So this is how we identified that CBP alpha, in our case, would need to be supplemented by Sanger sequencing. Now, we're not sequencing every sample that we get for CBP alpha, but we are restricting sequencing to those samples in which CBP alpha information would be critically useful. And so these are cases without any known bad prognostic markers, and so cases without things like FLT3 ITDs, for example, and which is a primary AML, would be Sanger verified to ensure that CBP alpha is adequately covered, and also that there are no mutations that we are missing by NGS. I'll move on to a second example here. ASXL1 there's a specific variant that occurs at C.1934, which is a duplication of a G. This occurs in a homopolymer region consisting of a run of 8 G nucleotides. And so this is again a hotspot mutation that occurs frequently in different myeloid malignancies. And we also wanted to understand in this case what is our minimal threshold for defining an ASXL1 positive sample. So in this case, in the bottom left-hand side, we have looked at all the samples that we'd sequenced up until this point when the analysis was done. It's about 500 cases, or slightly less than 500 cases. And you can see, based on the variant allele frequency detected at this particular site, the C.1934 site, that there is a background noise level that we're seeing just under 5%. And so this is an example where a mutation, for example, in this homopolymer site needs to be treated differently, in that you need to understand what the noise level is for your particular genomic region before defining what your positive sample is. And so in our case, we are calling everything that is above 20% VAF for this particular region as positive. Anything between 15 and 20% we are currently saying or verifying and validating before calling it as a positive here. So our numbers are about 6% of cases. You see that this particular variant occurs at higher allele frequencies between 15 and 35%, whereas the vast majority of other cases, the variant appears at very low allele frequencies, and this is typically because of slippage at our DNA polymerase that happens because of the homopolymer. I mentioned FLT3 internal tandem duplications before, and this is prognostically relevant in AML. Now, the challenge with FLT3 is that the insertion sizes can range up to 100 plus base pairs in length. And because of the limitations of the amplicon-based technology, where our amplicons, or Illumina's amplicons in this particular case, are around 200, 250 base pairs, it becomes difficult to amplify a region that contains a large insertion. And so we wanted to test using cases that we know are already positive for FLT3 ITDs, ranging from 24 to 90 base pairs. How many of these are we able to pick up using NGS, using this particular panel? And we saw that the performance for ITD was, as expected, was not great. We have 15 of 31 positive cases that we were able to pick up. So that is really not really effective for our sensitivity and specificity. So this, again, FLT3 ITDs, the detection here is performed using routine lab analysis on the other side, which in our routine lab, which is currently our FLP analysis. So I'd like to move on from showing you some of those examples to sample types and how different sample types perform in this assay. Like I mentioned, we expected to receive both blood and bone marrow tissue types as part of the analysis, and so we assessed both. We've looked at DNA extracted from both blood and bone marrow from paired blood and bone marrow from the same patient, and we looked at how they performed in terms of total coverage, number of reads, aligned reads, and reads on target. And as you can see from this graph here, there's really no significant difference between any of these parameters, and the two sample types performed equally well in the assay. We then went on to look at what variant types were actually detected in these cases. And this is data from about 30 paired cases that we've tested to look for what the actual variant profiles are between the blood and the bone marrow. And you can see in blue here are, in the majority of cases, in about 62% of cases, both blood and bone marrow samples had identical molecular profiles. We saw no differences in the two. In about a third of cases, you see bone marrow samples have a greater number of variants detected than the blood. And then in a small fraction of cases, you see either the blood having more variants, or that the two have different molecular profiles. And so that's expected if, based on tumor cellularity between the bone marrow and blood compartments, and also if sample collection is done at different times, which was the case in the last two, where the blood and the bone marrow were really different in terms of their molecular profiles. That can happen as you have tumor evolution happening between different collection times, and so that's the case. And so, so although it looks like technically they perform equally well in terms of understanding the biology of what's happening, the variant profiles are really reflective of what's happening at the biological level, either with tumor evolution or tumor cellularity differences between the two compartments. And so we prefer bone marrow in most cases as we do our analysis, but there are malignancies in which bone marrow taps are often difficult to get, such as in MDSs, and in those cases we profile the blood. So I'd like to now move on to our orthogonal verification methods. As part of our validation, we had Sanger tested all of the variants that came out from our validation data set, and we were happy with our Sanger confirmatory testing rates. And so as part of our routine test now, we only do Sanger verification in cases where we're unsure of the variant, either because of the alignment method or some other technical considerations that we look at. One example is when there's strand bias or read bias in detecting variants. This example is shown here, where all of the forward reads have one specific alignment, and all the reverse reads have a different alignment, and this, I believe, is in the CALR gene. So this is one example where we decided to do confirmatory testing to make sure what the actual alignment and what the actual nomenclature is for this particular insertion and deletion. Another example is end of amplicon reads, and you can see here an example of a deletion that occurs right at the end of the set of reads that are on the very top. Now this is oftentimes a trigger for us whenever we see a variant called at the very end of the read. That's something that we either need to verify before we report, or we need to see some stats that show that we're confident in this call. This particular variant was not Sanger. We had Sanger verified it, and it was not verified. And so end of amplicon calls typically are ones that we would need to verify prior to reporting. And my last example here is more complex variants. This is an example of a multi-allelic site where we see more than 2 variant alleles at the same nucleotide position. And in this case, it was also preceded by an insertion call. And so because of the number of different calls at this site, we had Sanger verified this particular call, and given that it's I think it was in the WT1 gene, and it becomes particularly more important when there is clinical significance associated with the gene of interest as well. And so our confirmatory testing guidelines in our particular lab would go on how technically confident are you in the call, and what is the level of clinical actionability that goes with that particular variant. Before I end this first section of my talk, I'd just like to also go through with you how we go through our variant filtering algorithm and our interpretation algorithm. What factors do we consider in calling a specific variant? And so this level of filtration within our data set happens within the Cartagena interface, which is what we use for variant filtering and as well as storing and assessment. All of the input variants that get into Cartagena is assessed for quality. Now the MySQL Reporter software system assesses for quality and comes out with a pass or no pass filter based on different quality measures that it looks at. We look at everything that has passed the MSR's quality filters. Oh, here.
Okay.
Yeah. Sorry. I hope the connection is back here. We have our list of what we call actionable hotspot variants. So these are variants that we've collected over the last 3 or 4 years of doing NGS analysis. These are clinically actionable variants in any disease site, and so we look at if the input variant list contains anything that we would call an actionable hotspot, and those are reviewed right away. We exclude anything that is present in global population frequencies with an MAF greater than 1%, and so we assess exact ESP, 1,000 genomes, and dbSNP, and if there are variants present at greater than 1% of the population, we're currently not reviewing those. And everything else we are reviewing if they meet a minimum variant allele frequency threshold of at least 5% and a coverage cutoff of 100x. And there are exceptions to the rules sometimes, like for CVP alpha, because we know that it is a poorly amplified region, we've brought that depth cutoff a little bit lower. And so for CVP alpha specifically, variants are reviewed if they meet a depth of coverage of at least 50x. So one thing that we do, because in myeloid malignancies, because it's a heme disorder, it really is sometimes challenging to find a matched normal sample to exclude polymorphisms and to exclude germline variants. Our approach to enriching for somatic variants really is to use the power that really is in these different databases, population databases such as dbSNP, exact, 1,000 genomes, ESP, and a combination of these. And so we've looked at which combination of these different data sets can allow us to exclude the maximum number of germline polymorphisms, and that is the combination of all of the above, except for dbSNP, I think. Yeah. So it's the exact 1,000 genomes phase 1, phase 3, and the ESP that we use in combination to exclude all of what we think would be known polymorphisms or rare polymorphisms. And of the total variants that we see per sample, there's about a median of 427 variants that go into the analysis, and after the different filtering methods that I indicated in the previous slide, we end up with a median of about 2 variants that gets assessed for about every case. And there's a range from about 0 to 10 variants for every case that we look at. So just to conclude my initial part of the validation, the considerations, we have established both the test performance characteristics as well as the limitations of our assay, and this was important really for taking this test into the clinic, because this allowed us to identify that we need some orthogonal testing methods like Sanger for complementing NGS analysis in regions such as CBP alpha, which is technically challenging to amplify, and also use routine lab methods for detecting and reporting in large insertions in FLT3. And how it's important to really evaluate recurrent variant calls and suspected artifacts, and this is around the example of ASXL1 that I indicated previously. We've shown that samples derived from both blood and bone marrow perform equally well in our analysis, and but the variant profiles can be unique to each sample type, and it may be different if sample collection it happens at different times, or if there's different cellularity associated with the two sample types. I briefly touched upon the importance of confirmatory testing, Sanger-based testing, for accurate nomenclature of selected insertion-deletion calls, particularly if they occur at the end of the amplicon, and also variant, the triage and assessment that we use in determining relevant calls. So we've used all of this information. I'd just like to really very quickly take you through the implementation process and how we've used this in the leukemia trial that is currently going on at the Princess Margaret Cancer Center. This is the Princess Margaret Advanced Genomics in Leukemia trial, the AGILE trial. This was launched 2 years ago now, in February of 2015. And it currently is recruiting all new AML, MDS, and MPN patients that come into the clinic weekly. And this is conducted, NGS testing here is conducted in parallel to standard molecular testing that these patients would normally get in the routine molecular diagnostics lab. And the results that we get are reported out to physicians at the end of the test. Just to show you what our case distribution currently for this trial looks like, almost half of our cases are AMLs, and the rest is comprised of MPNs, MDS, MPNs, and MDS alone. We have a few cases of lymphoid malignancies, and although the panel is not designed for testing lymphoid malignancies, it is really a myeloid specific panel. There is some information that you can get for lymphoid as well, and the clinicians are finding it of use in these cases as well. And so you can also see from the graph on the right-hand side that the vast majority of cases are positive for at least one variant. In most cases, over 90% of cases are informative for at least one variant in all of these different disease sites that we test for. The majority of the genes on our panel have been positive for at least one variant, and so you can see here some of the more commonly mutated genes towards the left-hand side, TET2, ASXLD, and MT3A. We've also looked at what types of mutations we're able to see in these genes, and this is also expected given the disease site that we're studying. For example, NPM1, the majority of variants that we see here are truncation mutations, and this is the result of the frame shift at codon 288. Oh, I think it's back on now. Apologize for that. Yes, as I was mentioning, truncation variants in NPM1, the majority of variants that we see in NPM1 are really this hotspot variant that we see. And so the variants that are coming out of this panel are what you would expect for myeloid malignancies. We also classify every variant that comes out for reporting is classified based on clinical actionability, and this is based on a classification scheme that we've developed in our lab and that's published here, Sukai et al. In 2015, Genomics of Medicine. We classify variants based on, as I mentioned, clinical actionability into classes 1 to 5. And so class 1 and class 2 variants are those that have clinical actionability, and when I say clinical actionability, it includes information that helps in the diagnosis, the prognosis, or the therapeutic targeting in these patients. And so this happens either in the same disease site that the variant is detected in, or in a different disease site. So these are class 1 and 2 are hotspot variants that are known in the literature to be actionable either in the disease type in which you identify it or in a different disease site. Class 3 and class 4 variants are not hotspot variants. They are variants in genes that are known to be actionable, but the specific variant itself may either be unknown or not have known significance. And class 5 variants are those in which actionability information is not yet available. And so if you look at the distribution of variant classes that we're seeing as part of this trial, almost a third of variants that we see here are actionable in the disease site in which we identify it. So about 33% are class 1, 5% are class 2. Another third of them are class 3As, which are you may not have information at the specific variant level, but you know that that gene is actionable in the disease site you investigated in. And so this is very clinically useful for the physician getting information and prioritizing them into the different classes based on what's known in the literature. And before I end, I'd just like to show you just an example of the complexity of the data that's coming out of these studies. And so this is a landscape map where each column really represents one patient. This is about 400 odd patients in the AML category. And here you can see along each row really represents one specific gene, and a colored box would represent a mutation in that specific gene. And so this just allows you to see how complex the data is that we're receiving, and also some of the co-mutation events that you can see, which genes are often mutated together in which disease sites. And we've also color-coded this based on actionability, where I think anything in red is not actionable, anything in blue is actionable. And so these are some of the ways in which we actually visualize the data. It does take some time to actually look through it and understand what's going on, but there are different ways in which different tools that we have to allow us to do this. And this is another way of representing that data. These are, instead of genes, you see networks or pathways, and that's allowing us to sort of collapse the data and look at the data in a different kind of way. Instead of asking which genes are mutated in these patients, we look at which pathways, and that hopefully give us some idea onto how to target and new ways to understand interactions and understand how to target patients based on their mutation profile. So this really brings me to the end of this discussion. Just to conclude, we see that the usefulness of NGS panels really in heme malignancies. We see clinically actionable variants that are coming out from these studies, and there's increased information benefit definitely from using single gene based testing to moving on to this parallel testing of multiple genes, and so that's something that we're able to clearly see from our analysis. And the identification of potentially clinically actionable variants, and this is something that is relevant to the physicians who are treating these patients, and that's something that we're getting as feedback from our studies, how to really look at and assess the variants that we see in terms of clinical management. That seems to be an important piece of this analysis as well. So none of this work would have been really possible without the entire lab. this is a combined team effort led by Suzanne Camareed and Dr. Tracy Stockley, who are directing the lab. We also have, as part of our variant interpretation group, a group of 4 postdocs. We're all involved in variant interpretation, and also all the technologists in the lab are responsible for really generating and looking through the technical aspects of each part of the testing that we do. And so a big thank you goes to all of them and our funding partners as well. So thank you very much.
So thank you, Dr. Thomas. Thank you for sharing this work and the information. So I just want to say participants can submit written questions by typing them in the webinar's control panel. And so let's see if we have any questions. I don't see any questions listed, but I just wanted to I actually have a question or two. So I think way back in the start you had mentioned that the lab does consider the threshold for coverage to be 500. And so how is this determined? I think this was asked before in one of the other talks as well, but how is 500 determined for us?
Yeah, in some ways it is really something that we started with our initial studies. That's something that was a threshold that we started in the lab. But I think the threshold really depends on what you want your VAF cutoff to be. So obviously with a lower variant allele frequency cutoff, you would want a higher coverage threshold to be able to call variants at such low frequencies. And so I think we started it as like an arbitrary number, but definitely this is something that is ongoing in the lab to try and figure out how we can tweak that to see can it be lower, or how we can further refine that.
Thank you. And then I have another question. And so this was a memorable panel for the technologists in the lab, because it wasn't just, validating the commercial panel. It was also attaching these minor validations, and we had to do it for the Sanger sequencing of the CBP alpha. That wasn't done in the lab, and now we're Sanger sequencing samples. And also we did a minor validation to detect large insertions and deletions on our MySeq, because we were relying on a custom script. So that alone we had to do 2 separate and minor validations. And so from the annotation aspect, because I know in the beginning when we started doing this, we relied heavily on commercial software, and then this, as we advanced our work, we then incorporated Cartagena, for example. So how was that incorporated later? Was that a separate validation, or was that, how was that incorporated?
Yeah, Cartagena was a different validation. It was very helpful because I think the Myeloid system in itself is really a useful system to validate a lot of these different questions, because you have a model system where there are these large insertions, and so that's a group of samples that you can test. And the fact that you have recurrent variants that are so well characterized in the literature, and you have a bank of samples that are positive, that's actually very invaluable to start off with a sample set where that these are the mutations, and these are potentially challenging mutations to detect, and can we detect them, and if not, what do we need to tweak to be able to detect them? I think that was really the power of our analysis, the fact that we had a really good sample set to start with. And definitely Cartagena brought together some of the limitations of each individual variant callers, the limitations that exist there. And definitely in terms of annotation, I think Cartagena's annotation really helped us fill in some of the gaps that existed in MSR as we were looking through the annotation software there, which was in Variant Studio. And just being up to date with your annotation sources, I think, was very important to be able to be complete in those annotation pieces. That was also very necessary.
Okay, and so I still don't see any questions being typed, but I have one final question for Dr. Thomas, and that is the Agile study that started in 2015. Is that still ongoing? I'm sorry if I missed. That's still ongoing?
That is still ongoing, yes. Initially, we thought it would be for 2 years, and we now have funding for it to go on for another year. We are hoping that that would be a routine test, and so that's something that we're hoping to implement in the routine lab, hopefully in the coming year or so.
Okay, well, thank you.
Michelle,
thank you.
this is Christy. There are a few questions in there. There are a couple of questions in there.
I thought I heard something. So, yes.
If you can't see them, I can read them out. Would you like me to do that?
Okay. We can't see them. I can't see them.
Okay, that's what I wondered. Okay, so the first one is...
Sure, that would be great. Thank you.
Okay. Did you aliquot the reagents and use over 2 to 3 runs? We found the kit worked the first time but failed after that. That's the first one.
Sorry,
Sorry, we didn't hear all of the question. Did I hear if we aliquoted reagents into separate runs?
Yes, over 2 to 3 runs.
Because it worked the first time. I believe this was a very robust assay in our hands, and it wasn't something that we had to troubleshoot in that regards. No, it was very robust, and we just picked it up with Illumina's protocols and went with it.
Okay. All right. The second question is, in the 61% of cases where marrow and blood have the same molecular profile, do you ever distinguish congenital from acquired findings with material from 72-hour PHA-stimulated cultures?
Sorry, Christy, do we have other questions? I apologize. This is I'm new to this. Might be some technology and connection here too.
Can you hear me?
I'm sorry if that was a question. We did not hear that.
All right. Well, I'll save these and I'll send them to Dr. Thomas, and I will have her answer those, and we'll share the answers with the group.
Okay, that would be great. Sorry.
Okay, that's no problem. All right. All right, thank you, Dr. Thomas.
Thank you very much.
Thank you.